Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Master the Balancing Act of Biologic Formulation Development with Uncle. My name is Georgina Wynne-Hughes and I'll be moderating today's presentation. I'm delighted to be joined by our guest speakers, Dorian van Lijsbetten, Senior Scientist, and Liane Seyrens, Pharmaceutical Development Manager from Eurofins, along with Andre Miller, Product Manager at Unchained Labs. In this webinar, our speakers will present a case study on Eurofins' formulation development of a fusion protein. They screen different excipients for their influence on stability to find an optimized formulation that balances thermal stability and minimizes the tendency of the analyte to aggregate. Following the presentation, we will have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these at any time to the left of your screen. Simply type your question into the box. Without further delay, I would like to hand over to our speakers, and I would like to thank them again for presenting for us today.
Thank you very much for this introduction, and thanks to everyone in the audience for joining our talk, Master the Balancing Act of Biologic Formulation Development with Uncle. My name is Andre. I am the Product Manager for Uncle here at Unchained Labs, and I will spend the next 10 minutes or so to introduce you to Uncle and how it can be used for biologic formulation development. I will then hand over to Liane and Doreen from Eurofins CDMO, who will take us through the development of a formulation they did. Formulation development is the art of balancing different aspects of protein stability, like melting and aggregation. Another balancing act is riding a tandem bike. Have you ever tried that? That's actually also a balancing act in more than one way. For one thing, both riders need to keep their balance sideways to not fall off, and they also need to coordinate pedaling and braking. I can tell you from my own experience, as a sharp turn was the end of one of my rides once, and we ended in the water. There was luckily no harm done. If you balance things nicely, like these happy parents on their side-by-side penny farthing, there's no need to fear that the baby is going to end in the river. Likewise, in formulation development, we need to balance different aspects of protein stability against each other to make sure our biologic does not flounder. When we talk about balance, stability balance for a protein, there are two aspects that we need to keep in mind. Unfolding of the protein and also aggregation stability. So there is an equilibrium between the native state of the protein and the unfolded state. This is conformational stability, but there's also aggregation that can happen for the natively folded protein and also for the unfolded protein. Colloidal stability, aggregation, conformational stability, unfolding. Both of these need to be kept in mind when we're developing the formulation of a biologic. Now, what has an influence on the stability of a protein? There are intrinsic factors and there are extrinsic factors. The extrinsic ones can be broken up into environmental and formulation factors. Environment, temperature, kinetic energy, is there shaking or not, and oxygen, this is one thing. And these are really not things that we can influence much. They are kind of a given. These are the ones that we have to optimize a formulation against. Intrinsic factors like the protein's primary structure and then an extension of that potential disulfides that will stabilize it are also not something we can influence unless we want to start a protein engineering framework project and change the primary structure of the protein. So the screw that we can turn on to influence the stability is formulation. And we can change the pH, we can change the salts, we can change carbohydrates, add some, remove some, sucrose, trehalose, to name two. And we can also work with organics, so detergents or proteins to stabilize our protein. And this whole thing, this development of a formulation becomes a balancing act because something that influences one of the stability aspects a certain way doesn't necessarily have the same effect on the other. I mean, how easy would it be if we could formulate everything in arginine and be done with it? But that doesn't work. Arginine works really well against aggregation, but it also is not good at preserving the native structure usually. So that's where the balancing act comes in. So when you want to test or when you want to find the best buffer, you really are going into an iterative process. You add your analyte, in this case I used the cartoon of IgG in red, to different buffers and you see how it performs. And then when you have some that you like, you refine these further by trying excipients. So it's a very iterative process to screen many different conditions and see which one works best and can be further optimized. So here in this case, we're looking at different melting temperatures. We find the best options. We look at the conditions that perform best and refine these further. It's always good to keep a couple of backups, and also we want to abandon some that we see do not perform the way we want, because you start with a large number of conditions and you want to whittle that field down to the promising candidates that you can then optimize further. How can you do this? What is a good tool to use for this work? Enter Uncle, the original all-in-one protein stability platform. Uncle combines fluorescence, static light scattering, dynamic light scattering in a temperature-controlled environment. For the fluorescence, we can record protein intrinsic fluorescence, or can excite protein intrinsic fluorescence, and we can excite dye fluorescence, for example, cipro-orange. We have two lasers to excite fluorescence, and we read the full spectrum emission between 250 and 720 nanometers. Static light scattering is a readout of molar mass, and the static light scattering signal will increase as the sample aggregates. So by following the SLS signal, we can say when is aggregation happening. I'll give you some concrete examples of fluorescence and SLS coming up. DLS is an orthogonal measure of aggregation. This looks at the diffusive properties and it gives you information about the hydrodynamic size of our analyte. So DLS is orthogonal to SLS. And because Lian and Dorian didn't use DLS in their work, I'm mentioning it here, telling you that it's in Uncle. If you need more information, please let me know. Happy to chat, but it's not going to come up in the rest of the talk. Temperature control is Uncle can be set between 15 degrees Celsius and 95 degrees Celsius. So flexibly usable for isothermal experiments, also for thermal ramps. These three measurement modalities and temperature control are coming together to form or to offer more than a dozen unique applications in Uncle. TMTAG, finding the melting and aggregation temperatures, is the one that we're going to be talking about in the second part of the talk. You can also use Uncle for a thermal flow or DSF experiment, so find out the melting temperature with a dye like cipro-orange. You can use Uncle for isothermal work, several days or several hours or days at a given temperature. You can use it to look at viral vectors and their payload loss. You can use it for DLS experiments. You can find out the colloidal stability parameters, KD, the fusion interaction parameter, the second viral coefficient, B22, and also Kirkwood buff, G22. So very flexible, over a dozen applications at your disposal. The magic in Uncle, the heart of Uncle, is the Uni, our sample holder. It's a black anodized aluminum frame with 16 9 microliter quartz cuvettes. You can see here they are open at both ends. That makes it easy to load them. You just have your sample in your single channel pipetter or multi-channel pipetter, bring the pipette tip close to the opening, and then capillary force will wick the sample right in. Of course, when you run your experiment, you don't want it to be open to the environment, so we clamp the Uni into the frame that has two silicone seals that press against the openings of the cuvette, and they prevent evaporation, so you don't have to worry about that for your thermal ramp or for your isothermal experiment over several days. Once your sample is in the Uni, you have to put it in Uncle, and the hardware interaction looks like this. We open the door, we remove the stray light cover, reveals the copper hot plate, that's the temperature controlled part. The Uni snaps in place by magnets. The anodized aluminum of the Uni contacts the copper, that leads to quick thermal equilibration. We replace the stray light cover, close the door, and then we can set up our experiment in the software. For a thermal ramp, the raw data looks like this. So we have a fluorescence emission spectrum for every degree of the ramp here in this case. That was the settings that we chose. You can see between 250 and 720 nanometers. And as the protein unfolds in a temperature gradient, the hydrophobic aromatic amino acid residues that are usually on the inside are coming into contact with the hydrophilic environment that leads to a decrease of the fluorescence, and most proteins also shifts to slightly longer wavelengths. So we can see that raw data here, and that raw data is then processed in Uncle software into a plot like this. So we have now on the left y-axis in blue, we have BCM, the barycentric mean. This is the spectral center of gravity of the fluorescence emission. It's a weighted average of over 120 wavelengths. leads to a high signal to low noise signal. And you can see that increasing here as the temperature increases. Then we have an inflection point and a second inflection point, the two melting events of our protein. What I want to point out also, Uncle determines the onset temperature. That is the temperature at which the fluorescent signal has deviated from baseline a certain percentage. And I want to point this out because Lilian and Dorian are going to mention this later. So from the fluorescence, we can see onset and the melting temperatures. Static light scattering gives us nice stable baseline. And then at around 61.5, 62 degrees, we find the aggregation starting, and that continues. Static light scattering and fluorescence are measured at the same time in the same volume and the same temperature, so we can directly compare, or we can directly put them into perspective to each other, so we can see here clearly that aggregation happens in concert with the second melting event. As I was mentioning, we measure the full spectrum of fluorescence emission between 250 and 720, so you can focus on the protein's intrinsic fluorescence. You can also focus on the fluorescence of a dye. If your buffer that you're working with, your formulation, quenches the protein's intrinsic fluorescence, you can use a dye, for example. And with that, I am concluding my intro to Uncle, our original all-in-one protein stability tool. It is flexible to be used for formulation work and to characterize protein candidates. You can focus on protein intrinsic fluorescence or signals from a reporter dye, depending on your needs. Uncle is also future-proof for a time when you work with viral vectors. There sure is an app for that. Now, I'd like to thank you for your attention and hand over to Lean to introduce us to Eurofins CDMO.
Thank you, Andre, for the introduction. And from my side as well, welcome to everyone who is attending. My name is Lien, and I am the head of the pharmaceutical development team at Eurofins CDMO in Ghent, which is in Belgium. My colleague Doreen will present this very interesting case study, but before that, I would briefly like to inform you on who we are and what we do. Eurofins CDMO is a contract development and manufacturing organization, and we offer services in drug substance and drug product development, both in human and in animal health, and for both small molecules and biologics. We provide support, development and manufacturing from preclinical development to clinical trial manufacturing up to phase 2B. And as we are a medium-sized CDMO, we provide high flexibility and we combine it with close communication adapted to the needs of small companies in the early stages of their projects. We have global capacities spread out over Europe, North America and India, which are focusing on drug substance and drug product development and manufacturing for both small organics and biologics. And our services, of course, include the required analytical support. The case study, which will be presented by Doreen, is situated in the drug product development for biologics and more specifically in formulation development for a protein. When it comes to drug product development, our formulation team in Ghent has expertise in drug substance characterization and formulation screening, in formulation development and in process development and scale-up. Our analytical teams are highly skilled in analytical method development, and we apply a quality by design approach in our development work, as will be presented in the following case study. When it comes to GMP manufacturing, we mainly focus on production of material for clinical trials in phases 1 and 2, and of course we provide the necessary regulatory support. Having introduced us, I now give the word to my colleague Doreen.
Thank you, Lien. Hi, my name is Doreen van Lijsbette. I'm senior scientist here at Eurofins CDMO, and I'm responsible for the formulation of the biologics. So today we're talking about the formulation development of a protein, and this protein is a fusion protein which contains 3 domains. Unfortunately, I cannot tell you more about this protein because this is confidential. What I can tell you is that we aim for a parenteral administration at 10 milligrams per ml. But to save material, we will do all the screenings with the uncle at 1 milligram per ml. Since the protein has 3 domains, we're expecting to see 2 to 3 melting points depending on the resolution. For the formulation development, we use a 2-step approach. So we have first the buffer screening, and then we decide on the best performing buffers, which we take along in the excipient screening. We also test for different tonifiers, stabilizers, and antioxidants in this excipient screening. Important to know is that we aim for an isoosmotic formulation, so the tonissifier will be crucial here. And we all do this using a design of experiment, which allows us to extract the most relevant conditions. So first, the buffer screening. We screen for 13 different buffers. These buffers are all allowed for parenteral administration, and we screen with from a pH 4.5 to 7.5. So we screen for citrate buffers, histidine, phosphate, acetate, histidine acetate, and tris buffers, and we all do this at a buffer strength of 25 millimolar. We also check for 3 extra conditions where there's some sodium chloride present. For this, we use the conditions histidine at pH 5.5, phosphate at pH 7, and acetate at pH 4.5, and there will all be 50 millimolar sodium chloride present. So here you see the first uncle results. On top we have the citrate buffer, and then on the bottom we have the histidine buffer. So on the citrate buffer, here on the left-hand side, this is the fluorescence signal. On the right-hand side, we can see the SLS signal. We can also see the vertical lines here. These are the melting points, and then on the other hand, we have also some vertical dashed lines here in the SLS signal. These are the aggregation points. So then if we look to the fluorescence signal, we have an increasing TM from blue over orange to green. So this is actually an increasing TM with a higher pH. If we look to the SLS signal, we have a lower aggregation point for the orange curve, but the green and the blue curve are quite alike. We can see an increasing in the SLS signal, and afterwards this signal is decreasing again. This is because the protein is precipitating. If we look back to the fluorescence signal, we can see these bumpy results here, and this is due to this precipitation. If we look to the histidine buffer, we can actually see 2 melting points here. So we have a first set of melting points here, and then a second set here. Again, we can see this increase with pH. So the red one is at pH 5, which has a lower TM than the blue one at pH 5.5, and the green one at pH 6.5. More interesting results here is in the SLS signal, where we can see this increase in SLS signal around 60 degrees for pH 6.5, while we don't see any aggregation or very small aggregation for the other pH values. Let's continue with some other buffers. So here on top we have the phosphate buffers and below the acetate buffer. We can see here for the phosphate buffer, we do have small differences, but no big differences in TMs are obtained. Again, here in the SLS signal, the aggregation points are very close and all around 60 degrees. Again, we have this high increase and again, some precipitation. Then for the acetate buffer, we again see a very interesting result between the different pH values. So at pH 4.5, the light green one, we have a lower TM, but then at pH 5.5. If we look to the SLS signal, again, this is very interesting. We have no aggregation at pH 4.5, but we do some aggregation at pH 5.5. We can also see almost no precipitation, so this is also very interesting. This is also seen in the fluorescence graph, where you can clearly see the 2 melting points here. Then the last 2 buffers is the histidine acetate buffer and the tris buffer. We can see quite a difference between those 2 buffers. So the tris buffer in green has a very low TM. The histidine acetate buffer is a higher TM at around 60 degrees in purple here. We mainly focus on the first TM because this TM is the most crucial for the stability of the protein. If you look to the SLS signal, you can see again here this aggregation in tris buffer, while we don't see aggregation in the histidine acetate buffer. We can also go to the conditions where we tested sodium chloride. So we can see here the difference in histidine, phosphate, and acetate buffer. Here we don't see any difference in the fluorescence signal in histidine buffer, but again, you can see quite a difference when we look to the SLS signal. So here we do see aggregation when sodium chloride is present. For the phosphates, there's some differences in the melting point. It's rather small, but we do see a lower TM when no sodium chloride is present. And also we can see a lower Tagg when no sodium chloride is present. So for the acetate here, we can also see a bigger difference. So the dark green one is the one where sodium chloride is present, and the light green where no sodium chloride is present. And again, we see this difference in the Tagg, where we can see an increase in SLS signal when sodium chloride is present. So here we know that in most of the conditions, addition of sodium chloride is not beneficial. I put here the rankings of the TMs here, so you can also see the actual values. So the TMs ranges from 63 to 52 degrees, and the Tagg ranking goes from 95, where we actually don't have any aggregation, to 55 degrees. On the left-hand side, you see the ranking according to the TM. On the other side, you can see the ranking of based on Tagg. I also put the top 4 of the other condition in orange here. And you can actually see that we do have this different rankings, so that the ranking is actually completely different if you look to TM and Tagg. So this will be very important. How do we choose now if we want to have the best conditions based on TM or the best conditions based on Tagg? So we have to look into our next steps of the formulation development. We can actually screen for conditions or from processes which have temperature stress or shear stress. So this can also have an impact on which melting point or aggregation point, which one we want to choose. We can also focus more on the long-term stability or the long-term storage of the formulation. And this can also depend on the application or administration type. So for this protein, we know it's prone to shear stress. So for that, we know that aggregation is quite an important issue. If we also look to the differences in TM and Tagg, I showed you a few or 2 conditions. You can see here that the difference between those 2 is 3 degrees in melting point, but in aggregation point, it's 34 degrees. So this is much larger. So therefore, if I go back to the ranking, we go for the best conditions ranked based on Tagg, but we do also take into account the TM ranking. So therefore, we go for the histidine acetate at pH 5, histidine at pH 5.5, and acetate at pH 4.5. Those are listed here. So these buffers we can take along in our excipient screening. So, as I already mentioned, this excipient screening is performed with the design of experiments. So we have different factors, 4 different factors. So first we have the buffers. Again, we screen at 25 millimolar concentration. Then we have the tonistifiers. For this, we choose some sugars, polyols, and also the sodium chloride. And we want to have a concentration of around 200 millimolar, but of course this is dependent on the osmolality contribution. And since this is higher for sodium chloride, we screen at a concentration of 100 millimolar for sodium chloride and 200 millimolar for sucrose, mannitol, and trehalose. The stabilizing excipients that we test are arginine, cyclodextrin, glycine, and also no extra addition of stabilizers. And this will be screened at 50 millimolar concentration. And then finally, we also screen for an antioxidant, and this is either EDTA, methionine, or no addition of antioxidant. And these are screened at 3 millimolar concentration. So in total, we have 96 experiments, which is actually a well-played setup. And we use a D-optimal design. And this design allows us to screen for a lot of experiments, but we don't have to combine all the different buffers with all the tonistifiers and stabilizing excipients and antioxidant. We can also extract the information on certain combinations without testing all the combinations. For this setup, we check for different responses, being the TM, the T-onset, which is the onset of the unfolding of the protein. We also check again the aggregation point and also the maximum SLS signal that we obtained during the run. So you can see here some of the Uncle plots. Again, we have the fluorescence signal on the left side and the SLS signal on the right side. Here you can also see, apart from the vertical full lines, you can also see some dashed lines, which is the T onset. So these ones are, again, as before, the TM. It's quite interesting here. So this blue curve, which is actually with sodium chloride. So here I have the three tonifiers shown. So it's trehalose, mannitol, and sodium chloride. And you can see a much lower melting point with sodium chloride, while the one with trehalose and mannitol are much higher. Also, we can see this both in the TMs and the T onsets. If we look to the aggregation, we have no aggregation when we have trehalose and mannitol, but we do see an increase or an aggregation when sodium chloride is present. Again, we have also a very high SLS signal. Then if we check, or I give you an example of the influence of the stabilizing agents. So I have an example here where we combine or we compare 50 millimolar arginine, cyclodextrin, or glycine. You can see here in blue that this is the worst TM or the lowest TM. This is an arginine, and that cyclodextrin and glycine have more or less the same melting point. If we look to the T onset, there are quite or smaller differences, but again, the arginine is the worst condition. If we look to the SLS signal, we see again aggregation in arginine and no aggregation or almost no aggregation when cyclodextrin or glycine is present. So I already mentioned we do a design of experiments, and we don't have to look at all the different conditions separately. We use a software for this. We can input all the data from the 96 experiments, and then we get a summary of fits, which actually tells you how well your model that you want to extract from these experiments, how well this is defined. So we have 4 parameters for this. They're all having a so the R square is telling how well your fit is, the Q square, how well your predictive fit is. We have also a model validity, which should be as high as possible. And then we also have the reproducibility. I also have to tell you that we have twice a reproducibility of 1 for the Tagg and the SLS max, and therefore the model validity cannot be calculated or is a negative value. If we look to the different models for TM, Tagg, SLS max, and T onset, we can see that the model for TM has very high values, and also the one for SLS has very high values. So those are the best models to look at. If we then look at the model, we can extract a coefficient plot, and this coefficient plot gives you in a bar graph the effects of the different factors. So we have here the melting point, and we want, of course, to have a higher melting point because a higher melting point is a higher stabilization. So we are looking for positive coefficients bar, and we can see it here for histidine and more pronounced for histidine acetate, and also sucrose, mannitol, and trehalose have a positive effect. The acetate buffer has a rather negative effect, and also sodium chloride and the next one, arginine, also have a negative effect. Then we continue with positive effects for glycine, no stabilizers, and EDTA and cyclodextrin. The EDTA, has a rather negative effect, and then methionine or no antioxidant or slightly positive. Then we can also check for different combinations. For example, here we have the combination of histidine with sucrose, and this is a rather negative effect. And we can continue for all the different combinations here. Sometimes we can also see that a certain combination is not significantly impacting the TM. So I listed all the combinations effects here, which are positive or negative, but it's mainly important to look at the single effects, let's say, which are presented here. We can also, as I mentioned, look to the model of the SLS max. So the higher the SLS signal, the more aggregation is there. So here we're actually looking for negative coefficients bar. We And you can see here that the acetate buffer, sucrose, mannitol, trehalose, cyclodextrin, these actually give the best results. And as we already noticed in the buffer screening, again, we have this different impacts of the different buffers on the TM and the SLS max. So here we have different values when you compare to TM. But for the other factors, tonifiers, stabilizers, and antioxidant, we do see similar positive effects for sucrose, trehalose, and mannitol, and rather negative effect on sodium chloride. We have a positive effect for cyclodextrin, glycine, or when no stabilizer is added, and a negative effect for arginine. Also, a more positive effect for methionine or no antioxidants, and negative effect for EDTA. So with these results, I come to the end of my slide deck. So to conclude, I hope you can see that we can have a big impact on TM or Tagg based on the pH and buffers that you use, and that we can see a difference in TM ranking and Tagg ranking. So it will be very important to balance this and choose for either a TM increase or a Tagg increase. For this protein, we decided to focus on the aggregation point, and we had the highest aggregation points in histidine, histidine acetate, and acetate. So we took these buffers along for excipient screening, where we show that sodium chloride and arginine have a negative effect, and that the best tonifier was trehalose, but also sucrose and mannitol were performing well. We saw the lowest aggregation in cyclodextrin, and we also had good results with methionine and either no antioxidants. So if you're wondering what the next steps are in our formulation development, we will use a confirmation of these different selected conditions, and we use them in a short stress study. So we stress these formulations up to 2 weeks at 25 degrees and 40 degrees, and we test the stress samples with a broad range of analysis techniques. This was the end of my slides. I hope you enjoyed the results and the possibilities that you have when screening with the uncle. I thank you for listening, and if you have any questions, you can.
Thank you for that very informative presentation, Dorian, Leanne, and Andre. So it's now time for our question and answer session. So if you have any further questions, remember you can still submit these, and you can do this by just typing in your question in the box to the left of your screen. So our first question we have, I think this is for Andre. So what is needed to run Uncle other than Uncle itself?
Thanks for sending in that question. Uncle requires the uni that I showed you earlier in one of my slides. So it's our sample holder. It's an array of 16 quartz cuvettes that have a volume of 9 microliters each. That is the consumable that is required to run Uncle. The frame with the silicone gaskets I also showed, that would be something that you can reuse, the gaskets, and the gaskets themselves need to be exchanged. The uni is the consumable you need, and you can use them partially, so you can use a couple of the wells one day and the rest on the other day. Other than that, in the Uncle, that's all you need, a computer with the software, but that we provide as well.
Thank you. And a question here, I think, for Dorian and Leanne. So how long did this study take?
Okay, this study, we performed it in 2 steps. So the buffer screening, if I include all preparations of buffers as well, it would take around 2 days to perform, including data analysis. And then if we go to the excipient screening, of course, we have much more experiments there. We do have some more excipients that we tested. So I think this would take around 3 to 4 days, if I remember well. So in total, within 1 to 2 weeks, you will have a very broad screening of a lot of excipients, and you can continue easily your formulation development.
Thank you. And another question for you. So would you call a study of this size small, average, or large?
I think it's more on average or rather large study, let's say. we tested, especially in the excipient screening, we tested for 96 experiments. This is already quite a lot. You also get these 96 graphs from the uncle, so it will take some time to do the data analysis and check all techniques. But then, of course, we use the software of the design of experiments to analyze all the data and to see the best results in the best conditions. So it's a bit bigger study, but of course it will give you a lot of information which you can use afterwards.
Thank you. And another question for Andre. So what about using Uncle in a GXP environment?
Uncle can be used in a GXP, so good manufacturing practice environment. We can qualify the instrument, so do installation and operation qualification, and also our software has a 21 CFR Part 11 mode that allows the audit trail to be on. So Uncle is ready for a GXP environment.
Great, thank you. And another question for you, Andre. So how long does a thermal ramp in Uncle take?
That's an interesting one. Thermal ramps in Uncle depend, of course, on the number of samples you use and the ramping speed you choose. So you as the user, as the experiment designer, have the full flexibility over that. You can decide how fast you want to go, how many reads you want to do per degree of your samples. But I think a good rule of thumb is about 2 to 2.5 hours is for a thermal ramp.
Thank you. And another question here for Dorian and Leon. So how do you make the Uncle data readable for other software like D-Optimal Design that you mentioned?
Actually, it's quite easy. So you can export the Uncle data to Excel, and then you can basically input it in a lot of different softwares. It's also quite easy to do the data analysis because the Uncle software already checks where the TM, TX, the onsets are. So it makes your life a bit easier because you only have to check if the software did a good job there and had the right melting point at the right place. And then afterwards, you can just export to Excel, and then we imported this Excel file into our design of experiment software.
That's great, thank you. And another question that's just come in. So can Uncle be used for small molecules, for example, oligopeptides, or only for proteins?
Thanks for sending that one. An interesting question. The Uncle can in principle be used not only for proteins, but also for smaller ones. The requirement, if you want to look at their fluorescence, is that they need to fluoresce. So the smaller peptide or oligopeptide would have to have a residue, an aromatic residue in it. And then we can take a look, of course, at the change of fluorescence as a function of time or temperature. If the small peptide does not have that, so if there is nothing fluorescing in there, then you can use a reporter dye. That's the nice thing with Uncle, is the flexibility of being able to use both the intrinsic fluorescence and also extrinsic dye, reporter dye fluorescence. So in principle, there's nothing limiting you use in Uncle for either system. So as long as you have a fluorescent signal that you can monitor, the change of which you can monitor, you're good to go.
And maybe if I can add to that, it's also interesting if you just want to follow aggregation, or you can also follow the DLS signal at a higher or lower temperature. And then you can also check how your oligopeptide or your smaller protein is behaving.
That's great, thank you. And I think we have time for just one more question. So we'll finish on how to best contact Unchained Labs and Eurofins.
Okay, let me answer that one because I think our audio stream was cut slightly short there. So if you want to get in touch with Eurofins, I think www.eurofins.com/CDMO is where you can find their contact info. And if you want to talk with us here at Unchained, it would be info@unchainedlabs.com. And then from there, we'll get back to you and can start the chat.
Great, thank you. And just one final question. Can Uncle be used for very high concentration proteins like 400 milligrams per ml?
Yes, it can. We have done work with very high protein concentrations. I think I've seen data set that went up 300, 350 milligrams per milliliter. So in principle, Uncle's dynamic range is very high. So I don't see anything speaking against using it there. The good thing is there's no microfluidics. There's nothing that could, where the viscosity of those high concentrations could be prohibitive to using it with our uni. So that should be good. And whoever asked that question, send us an email and we can continue talking about this. And I can provide you with some example data for very high IgG type molecule concentrations.
That's great. Thank you very much. So that's all we have time for today. And I would like to thank our expert speakers once again for today's informative presentation and discussion. And a big thank you to everyone joining us online. We hope you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net and I will follow up with your questions for our speakers. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.